Hi everyone, good afternoon. Welcome to our short and special webinar session. This event is brought to you by students of Biomedical Science, Faculty of Health Sciences, Universiti Selangor. My name is Marcus Young from BMS 17, MC of the program today. Ladies and gentlemen, we have a special webinar topic today entitled DNA Fingerprinting, How It Works. This topic will be delivered by Dr. Muhammad Nazif Samad, one of our lecturers in the Faculty of Health Sciences, UNISEL. Before I invite Dr. Muhammad Nazif, I would like to make an important announcement. By joining this webinar, the audience will have a chance to grab your e-certificate. How to get the e-certificate? Please visit fsk.unisel.edu.my/events after this webinar session. Then, fill up a very simple quiz there and submit it before 11:30 p.m. tonight. Ladies and gentlemen, so without further ado, I would like to invite Dr. Muhammad Nazif to deliver our interesting topic. With all due respect, Doctor, welcome.
Okay, thank you so much, Mr. Marcus, our MC of the event today. Assalamualaikum, salam sejahtera to all ladies and gentlemen. Welcome to a short webinar session. And the title is very interesting, especially for those who wants to know, who wants to understand further on the uniqueness of ourselves. Okay, this topic will be great to be elaborated by showing some pictures and some animated graphics to help us easy to understand on how DNA fingerprinting works. Okay, let me share the screen first. Okay, wait. Okay. Today webinar is actually one of the topics that learned by biomedical students through the lecture session, and we try to bring a bit different way of conveying the lecture content. We use different platform, the FB Live, so we can share to more people regarding one of exciting topics that they learn in class. Okay, first of all is about fingerprint. We can find a mark on the surface of our fingertip, and the mark comes with unique pattern, and the pattern able to be used for identifying individual. So the unique pattern of lines on the fingertips here, such as arch, loop, okay, allow the identification purpose, as it is very unlikely that any two people would have exactly the same physical fingerprint, and this gives huge benefit to human through biometric technology to allow only selected person to be authorized to access certain information, such as recording of certain information such as attendance and so on. Then we go to another term. It is DNA fingerprinting. Okay, actually, fingerprint and DNA fingerprinting are two different things. Okay. However, people or scientists include the fingerprint term in DNA fingerprinting because it is very unlikely that any two people would have exactly the same DNA information in the same way that it is very unlikely that any two people would have precisely the identical physical fingerprint. And DNA fingerprint technology has evolved from a simple analysis using gel. Okay, then it comes with the aid of computational analysis using certain software and high technology instrumentation to provide comprehensive, fast, easy, and more accurate results. Okay, before we go further on how the DNA fingerprinting works and how the analysis is made, it is important to know the purpose of performing the test. So DNA fingerprinting is useful for finding out who a person's parents or siblings are. So this test also may be used to identify the parents of babies who were switched at birth. Then the test also become an important tool to solve crime. Yes? Samples such as blood, semen, skin, or other tissue that left at the scene of a crime can be analyzed to help prove whether the suspect was or was not at the crime scene. Then the test also important to identify a body. As example, if the body is badly decomposed or if only body parts are available, such as following a natural disaster or a battle or other situations such as MH17 aircraft that was shot down in 2014. Okay, in terms of history, the DNA fingerprinting was developed in 1984 by a British geneticist, Professor Sir Alex Jeffrey. Okay, and then from his observation, he noticed that certain sequences in human genome are highly variable DNA. So these sequences are called DNA satellites. Okay, DNA satellites consist of repeating DNA sequence, repeating DNA sequence, and he recognized that each individual has a unique pattern of DNA satellites, in which the number of repeats is highly variable among people except for monozygotes, twin. And according to him, the path that led to the discovery of genetic fingerprinting began when his father gave him a chemistry set and a microscope. Okay, when he was at 8 years old, his father had instilled his interest in science since he was a child, and his father gave him a real chemistry set, not a child toy. And later, when he was 13 years old, he got a face full of sulfuric acid. That's why he wears the beard until now, and his current age is 72 years old. Now we go to the technique. Okay, simply put, DNA fingerprinting can be explained through this picture. Okay, we have a pattern of DNA. Okay, after analyzing sample obtained from a crime scene, as example, then samples from suspects are also analyzed and give this pattern. Then we can know from the scientific evidence it matched with suspect 2 here. Okay, and and this will aid in the process of investigation by the authorities. Okay, if we compare my DNA with my own DNA, of course it will be 100% similar because we are comparing the same sample. Then if we compare my DNA with my friend's DNA, Mr. Amir, we are same species, of course, Homo sapien. Okay, it is around 99.9% of our DNA is similar. Then with comparing of my DNA with other species, monkey, okay, it is around 98% of DNA is similar. Then with mouse, it is around 75% of our DNA is similar. So comparing Homo sapiens DNA with other species, showing that most part of DNA are same, as our system generally are same, means the foundation of the system, such as we have respiratory system, we have circulatory system, digestive system, and so on. And among us, okay, everyone is unique by around 0.1%, as humans are around 99.9% genetically identical, and only 0.1% of the genetic differ. Then the small portion of the difference, okay, this small portion of the difference can be employed for the identification of people based on the DNA information. Okay, let's we make a bit revision on foundation of genetic by looking back the level of genetics. Okay, firstly, we focus on our cell, the functional unit of an organism. So inside of cell, we can find nucleus, okay, consisting 46 chromosome. Human, we have 46 chromosome. Okay, they store important information. They are the blueprints of life. Then we zoom to one of the chromosome. Okay, we see what is inside of a chromosome here. It is a thread-like structure, okay, consisting of tightly coiled DNA. Okay, and when we zoom further inside of chromosome, okay, it includes packaging proteins called histone protein. Okay, and aided by other type of protein bind to and condense the DNA molecule to maintain its integrity. Okay, zooming to the next level, we can find the DNA strand. It is in double helix form. Double helix form. Okay. DNA or deoxyribonucleic acid is a long molecule that contains our unique genetic code. Okay, it is long strand consisting information. It is like a recipe book. It holds the instruction for making all the proteins in our body. And this is an amazing fact about our DNA strand. Okay, the length of each human cell's DNA is around 3 meters. The length of each human cell's DNA is around 3 meters. Each person has around 37.4 trillion of cell in the body. So, if the DNA strands from one person were laid out in a line, so the strands would reach the sun and back around 70 times. Okay, this is truly amazing facts about our DNA. Okay, then inside the strand, certain segments are genes. Okay, genes are a special recipe to give instruction for making specific protein. As example, for making our skin color, for making enzymes for digestion, hemoglobin for oxygen transportation, antibody for immunity, and so on. So skin color, melanin, the enzymes, hemoglobin, antibody. There is as examples of protein exist in our body. And we have around 25,000 of gene in our body. Okay, these three, gene A, gene B, gene C here are just example. Actually, we have around 25,000 of genes in our body. And besides the gene, most part of the DNA strand are classified as non-coding region. Okay, so non-coding regions are not translated into amino acid of protein, but they play a role in the control of gene activity, meaning they help determine when and where certain genes are turned on or turned off, and they are also important for protein assembly process. Our genome is largely consists of non-coding region. Okay, so more than 98% of the DNA strand are non-coding region, and less than 2% are coding region. Okay. So in non-coding regions, actually we can find many repetitive sequences of DNA. Okay, as example, the repetitive sequence is short tandem repeats, or STR, short tandem repeats. Okay. So as example here, agat AG80 here is repeated for 7 times here. 1, 2, 3, 4, 5, 6, 7. And gata here is repeated 4 times. So these are example of repetitive sequence that we call as STR, short tandem repeat. Okay, let's say this DNA is belong to Upin. Okay, then we also have other samples here, Mutu and Wang's DNA sample. Okay, so in the part of non-coding region, they are short tandem repeat or STR. Okay, so in this example, it is shown two sides of STR here, the green one and the blue one. Okay, every person has unique number of the DNA repeat. Example, Upin here got 9 repeat, but Mutu has 13, Wang has 4 for the first STR site. And here, this is the second example. Here we have 2 site of STR. Here we have 11 for Upin, 6 for Mutu, and 9 for Wang. Okay. So in DNA fingerprinting procedure, the STR site here can be cut. Okay, it can be cut using special enzyme. Okay, we call as rescission enzyme. Okay, we can cut here, can cut here, we can cut here. Okay, and then what happened after we cut or after we digest using the enzyme, we can separate them using electrophoresis lah. Okay, we got it. So electrophoresis will separate the STR site that we have cut. Okay, and the separation will be based on the size of the fragment. Okay, now we try to separate them. Okay, so the separation will be based on the size of the fragment or based on the molecular weight. The shorter fragment consisting lower molecular weight will be moving faster. Okay, as example number 4 here of Wang and number 6 here for Mutu. Okay, and the longer fragment consisting higher molecular weight will be moving later. Okay, in this case, example from Mutu, we have 13, right? The repeat is 13, so it will be moving later. So this fragment actually can be visualized under UV light, and it will appear as DNA band with certain pattern. So because of every person has a unique number of STR, so this will produce a unique pattern of DNA band which allow the identification of people. Okay, this is another example of the fingerprinting process. Okay, we have 3 suspects here, Mr. Nobi, Laila and Jeffer. Okay, so their samples were analyzed and compared with sample that we found at the crime scene. So we do the electrophoresis to separate the fragment. Okay, so the sample is matched with Mr. Nobi sample. Okay, so what happened after that? Further investigation can be done by the authority to solve this issue or this mystery. Okay. Okay, we are talking about satellites DNA just now. Okay, satellite DNA. Okay, DNA satellite consists of very large array of randomly repeating, and it is located in non-coding DNA. Okay, And beside STR, beside short tandem repeat, another type of DNA satellite in our genome is VNTR. VNTR stands for variable tandem of tandem repeat. And how they are different? Okay, they are different in terms of the size base pair and the size of repeat, in which VNTR is larger compared to STR. So that's why the STR is classified as microsatellite. Okay, and VNTR is classified as mini satellite. Okay. In DNA fingerprinting, both STR and VNTR can be used in the procedure of DNA fingerprinting, and from my reading, the scientists prefer to use STR because of shorter fragment. It has advantage to be analyzed using certain DNA fingerprinting technology platform. Okay, here we are going to talk a bit about samples that can be used for the DNA fingerprinting test. We can use buccal swab. We can use buccal cell. Okay, when we do swab on the cheek, in the mouth. Okay, actually we are collecting cell on that surface, okay, using special brush. The cells consist of DNA and can be used for genetic testing. Saliva also can give DNA. Saliva also can give DNA, as in the saliva we can get DNA from white blood cell and buccal cell that present in the solution in the saliva. And from blood, okay, also we can get DNA, okay, from semen, okay, from any tissue, okay, from hair, okay. But for hair, okay, it is important to use hair with the root or the follicle, as only the part consists of cell that bring the DNA. Okay, that's why it is important to get the root of the hair. Okay. Okay, then. Okay, after collecting the sample, DNA in the cell need to be extracted. Okay, and then need to be purified. Okay, and there are actually two common way to do the DNA fingerprinting. The first one is through RFLP, RFLP, or restriction fragment length polymorphism. It is a traditional method actually. It is a procedure by using certain enzyme, okay, to digest the sample. Then the fragments are separated using electrophoresis. Then the fragments are transferred to the nitrocellulose paper to perform the Southern blotting. Southern blotting. Probe hybridization is done using the radio label probe. Then it is exposed to an X-ray film to get result. Okay. And another way is by using PCR. Okay, everybody know PCR now. Okay, polymerase chain reaction. The PCR-based DNA fingerprinting technique is the most popular among all. This is the most popular. it is simpler than RFLP autoradiography, and PCR based method give reliable result compared to traditional techniques. Okay, using PCR, okay, the specific primer, we call as a primer, will amplify the DNA. Then the PCR product will be separated using gel electrophoresis. Okay, so a simple agarose gel setup is enough to get the result. So however, PCR based agarose gel electrophoresis is mostly used for VNTR analysis. Okay, because VNTR are larger than STR and it can separate properly. Okay, STRs are smaller in length, so it is difficult to distinguish in an agarose gel. So in contrast, VNTR are larger fragment and it will give a beautiful distinct band pattern in agarose gel electrophoresis. So then DNA sequencing, okay, also can be done for the confirmation of the amplified segment. Then computer application can be used to assist the analysis. Okay, as example, the result of analysis here is obtained by using fluorescent signal produced by reaction using certain DNA probe. Okay, so then the signal is detected by the computer using certain software. Then the peak of STR can be plotted and displayed by the computer. Okay, this is 13, 16, 28, 29. Here is the number of repeat of the STR. Okay. Okay. Let we try to see some of example of laboratory report of DNA fingerprinting. Okay, the first one here, okay, the first case here, two sample A and sample B here. Okay, sample A is from the crime scene and sample B is for a suspect from the suspect. Okay, here are loci. This is the loci or STR site. STR site. We have in this case STR site investigated in the DNA fingerprinting test got 23 loci, 23 STR sites being tested. Okay, in example just now, Upin, from the Upin right now, right, Mutu and Wang, we are utilizing only 2 STR site. Okay, in this case, in the real one, normally we use 23 STR site. Okay, and we can see here all of the STR number or repeat number are same. Okay, this one 18, this one 18, this one 12, this one 12, this one 19, this one is also 19. Okay, so here it is very strong to say sample that we found at the crime scene is most probably belong to the suspect. Okay. Okay, the second case here is other crime scene. Okay, sample A. Okay, sample A is from the crime scene, and sample B is from the suspect. Okay, here are the loci, okay, or STR site investigated in the DNA fingerprinting test. 23 loci or 23 STR site. Okay, so here it is very strong to say sample that we found at the crime scene does not belong to the suspect. This one 18, 16, this one 10, this one 10. Yes, this one same, but most of them are different. Okay. Okay, we move to the last case example here. In case number 3, here is paternity test to know relationship between two males. We have male 1 and male 2. Okay, 23 loci or 23 STR site being tested, and we can see here most of the STR number or repeat number are same. Okay, only 2 loci, okay, do not match between the sample. So as a conclusion, okay, we cannot exclude male 1 and male 2 from being of the same lineage. Okay, so after performing some statistical calculation from this result, okay, male 1 is 6 times more likely to be patrilineally related to male 2. So the specific way on how this 6 times chance can be calculated is we need other session to see how it is determined. Okay, the last one is about the service of DNA fingerprinting that offer in our country, and I know one company that give the test service located in PJ Petaling Jaya, Easy DNA, Easy DNA. Okay, and we need to know, and if we need to know more about it, okay, maybe we want to know the price, I mean we want to know the type of DNA test available and so on, we can get the information from the website easydna.my. Okay, and bahagian forensik DNA Jabatan Kimia Malaysia also has this service. Okay, Jabatan Kimia, as we know, it is under Ministry of Science, Technology and Innovation, MOSTI. Okay, I think that's all from me, and I hope this sharing is good for our knowledge, and also appreciate the uniqueness that everyone has. Okay, thank you so much.
Thank you so much, Dr. Muhammad Nazif, for your great sharing about DNA fingerprinting. Now we get a clear idea on how the technique works in analyzing samples to see their similarity. Ladies and gentlemen, we have a limited time for the Q&A session. If you have any question regarding our topic, please leave your comment and our speaker will make his response there. So now, ladies and gentlemen, get your e-certificate by visiting fsk.unisel.edu.my/events. Then fill up a very simple quiz there and submit before 11:30 p.m. tonight. Thank you, Dr. Muhammad Nazif. Thank you to all audience. Until we meet again, stay safe, everyone. I'm Marcus Young signing out. Have a great day ahead. Thank you.
Okay, thank you.